that support it, that's all. However, the two major branches within it, or actually we have three branches, one here, one there. We can't talk about them. So one of them, the biggest one, is chrome alveolata. Okay, chrome alveolata, which then has two branches, alveolata, straminopiala. Okay, so this here, and then another one that's further down, rhizaria. Okay, so these are the major ones for chrome alveolata. Okay, dealing with it. This here, the best apomorphy is that most of them that have chloroplasts. Now, many of them have secondarily lost their chloroplast, but we know that they came from an ancestry with chloroplasts because some of them have organelles called plastids. They're not photosynthetic. Some of them still have chloroplast genes in their nucleus. But all throughout the group, and we're not looking at every single, there's a lot of different groups here. We're just going to look at the major groups in here. But the loss of the chloroplast has happened multiple times. One of the problems with lost characters is that sometimes they look the same as if they did have a character to begin with. And so lost characters are not always good indicators of relationship. But in this case, if they have chloroplast genes, then they had to have had chloroplast. Okay, so there's a little bit of evidence there. Okay, so this is from the red lineage. So all of the chloroplasts in the photosynthetic organisms within this clade have chlorophyll A and chlorophyll C. Okay, in there, and usually the chloroplasts have 3 or 4 membranes indicative of a endosymbiotic event. Okay, and some of them, as I said, they have plastid-like organelles. Plastids come from chloroplasts. Okay, very similar. Two branches here, alveolata, straminopiala. Okay, straminopiala, you'll also see it called chromista, but for the straminopiala, it isn't two flagella. Two flagella is widespread, but the opposite apomorphic here is the structure of those two flagella. One is long and tensile. Okay. Now we also still find some organisms that have two flagella, one flagellum long, but not tensile. This here, they have cytoplasmic extensions off of the side of it. So these would look like you have a cell and one short, one long flagellum, and there's these cytoplasmic extensions all around it, and that's what we mean by tensile. Okay, and that's pretty unique. All of them have it. Okay, and it's one of the best features to diagnose this particular group. All right. So, furthermore, something else in their chloroplasts, they have a particular type of, in addition to chlorophyll A, chlorophyll B, they also have a particular type of pigment called glucoxanthin. Okay, this is for photosynthesis. So, as I said, since they come from the red lineage, they have chlorophylls A and C. Those that are photosynthetic. Okay, secondary endosymbiotic event. And once I say this, that it arose right here, then you should be, one of the beauties about knowing phylogenies is that if it happened here, everyone has it in one form or another. Okay, so it's a derived character for the them. So even though I'm saying this here, they have chlorophylls A and C, they have 3 or 4 chloroplasts, 3 or 4 membranes, and so forth. You don't have to memorize that for that. It all happened here, and therefore they all should have some form of that. Okay, that's the beauties of phylogenies. So 2 major groups here, or the major groups in looking at straminopiala. Okay, is that Bacillariophyta? You just need to know as diatoms. I love seeing that word. Okay, seeing it written. Okay, Bacillariophyta. But I'm making it simpler for you guys. You just need to call them diatoms. All right. So all of these are unicellular, and also Phaeophyta. You just need to know them as brown algae. They're multicellular. Okay, and they multicellularity has evolved multiple times in eukaryotes. This is independent of multicellularity in fungi and in animals, right? Brown algae. And the other one is oomycota. Used to be classified as fungi, common name water molds, but we still have a group of fungi that are still called water molds. And so a lot of times common names are inadequate. They're ambiguous, okay? So these are the three major groups of straminophilans. So the diatoms, they're all unicellular, most of them, a lot of them in fresh water. And the major thing, apomorphy for them, is they have cell walls of silicates, glass. So their cell walls have glass, and that gives them a glassy appearance. Also, you can also count this as a separate synapomorphy, if you will, is that the cell walls come in two halves, like the lid of a box, okay? One half is the bottom, one half is the lid, okay? And when they divide right along that line, and the one daughter cell gets the bottom of the box and secretes a new lid, and the other one gets the lid and secretes a new bottom, okay? And that's how they grow. But they have all these wonderful forms, and furthermore, in ancient seas that had a lot of diatoms, when they die, their cell walls float down and become part of the sediment. Therefore, you have all this glassy silicate material in that sediment layer, and that sediment layer is often harvested. It's called diatomaceous earth. They put it as an abrasive in scouring powders. In toothpaste, you can find diatomaceous earth because it will grind, right? These have diploid life cycles, okay? And for a lot of the life cycles of these, I was going to include them in this lecture, but I'm going to throw a lot of them out, okay? Because we just don't have the time. We've got to move on. All right, so these are menagerie of diatoms. They're all glassy-like. They have suture right here. So this is the lid. Actually, that's the lid. That's the box. And that suture is what's dividing the two halves of the cell. And they have all these little striations on them that give them a very interesting appearance. But they're all unicellular. Yes?
What's like the reason for their like varied appearance?
Well, it's because of transport vesicles that bring the material up to the surface and deposit it there. So it's a lot of times a pattern of how this material is made in the smooth replicular transport Golgi to the plasma membrane, and certain enzymes that are there that are also involved, and we have these repeating patterns. Okay, and those patterns differ from one species to the next. Okay, and that's what's responsible for that. All right. Phaeophyta. Here you just need to know them as brown algae. That's all. So apomorphy for these would be, you could say multicellularity. Okay. However, as I said, multicellularity is involved in eukaryotes more than once. But this is independent. They contain cellulose in their cell walls, but also a compound, which is a polysaccharide called algin. And so this is commercially important. So these are multicellular seaweeds, and I'm not going to impose this on you. This is a type of starch. So their excess photosynthate, they store it as laminarin starch. Okay, so it's sort of a starchy compound. And the whole body, we don't call these plants. They're not plants, okay? They're plant-like, but they're not plants, all right? So the whole body is called a thallus. So if it's not plant, and it's a big photosynthetic multicellular thing, we refer to the body as a thallus, all right? And so that thallus consists of, we find these in intertidal zones. You don't find them out in the deep ocean because they're photosynthetic. They have to be where there's light. So they have to be in shallow water where light penetrates down. Okay, further in the middle of the ocean, light is, it disappears in the upper 100 meters or so. Now out there, you can have diatoms because they float on the surface, okay? but these have to be pretty close to shore. So they're in an area where they are whipped back and forth by wave action. And therefore, to stay in one place, they have, it's not a root, but it's called a holdfast that anchors the thallus so that it doesn't get washed away, okay? And then they have blade or sort of branch-like things called stipes and things that broad surface areas called blades for photosynthesis. But in this case, both the stipes and the blades are photosynthetic, okay? And then to keep this up floating, instead of it falling down by gravity, they will have gas bladders, okay, that are filled with gas that keeps the blades suspended up in the water column, okay? So this is typical of these types of critters, okay? Now this alvin, it is commercially important because we put it in food, okay, because it can make it more viscous, okay, such as pudding. It's also used in auger plates, and also agarose, our gels are derived from this as well, okay? So commercially it's important. Olmycota, okay, as I said, this used to be classified as fungi, but we now know that they're not. They're called water molds or downy mildews. They are heterotrophic, okay? The other two groups were photosynthetic, okay? But these do have plastids. They do have plastids, but they're non-photosynthetic, okay? And so they've lost their chloroplasts. However, and we know that they belong to Stramnophylla because their reproductive cells that are sometimes called zoospores, they are, okay, these are filamentous, okay, sort of look like filamentous algae, all right? It's little square cells that are hooked end to end like a string. That's why they thought they once upon a time were fungi, and they're heterotrophic. However, they have flagellated zoospores, single cells for reproduction, and these have the same type of general stramnopyle flagella. One short, one long, and tinsel, okay? And therefore we know that they belong here. Now, the importance of these is that some of these are agricultural pests, okay? Some of them live in water, but some of them live on plants. For example, something called Phytophthora infestans, it caused potato blight, okay? Potatoes originated in the Andes Mountains in South America, and then when Europeans came along, they took them to Europe, and therefore, like in Ireland during the 19th century, that's all that they grew. And with Phytophthora, it destroyed in 1844, 49, completely devastated the potato crop. Millions of people starved. It was during that time also that you had heavy migration from Ireland to the United States, and to New York and New Jersey and those areas. But this is still around. How many of you have heard of sudden oak death? No? Here in California, you guys don't pay attention to the oak trees? My goodness. You guys don't get out much. Anyway, it's a member of this group that's causing a lot of the oak trees to die as well. Okay, so that's Stramnophylla. 3 groups. You just need to know brown algae, diatoms, and oomycetes, okay? The other branch, alveolata, also has 3 things. Major apomorphia of it is under the plasma membrane. They have these membranous sacs that are called, guess what? Alveoli. Should make it easy to understand, know the name of the group. Alveoli, and the apomorphy is, alveolata, and the apomorphy is alveoli. Dinoflagellates, ciliates, and apicomplexan. Unfortunately, I don't have a common name for apicomplexan, so that's going to be a name that you have to know. Just these 3 groups, all right? So dinoflagellates, they're all unicellular, okay? Mostly marine, okay? They're photosynthetic and have 2 flagella. What's significant about them is that they have these grooves, two grooves, transverse groove and another groove going perpendicular to it, and the flagella are inside of those grooves. Two flagella, and when they beat, usually these things are sort of tumble, okay, as they move through the water. Photosynthetic, you saw some of these yesterday on prepared slides. Underneath the plasma membrane, they have in the alveoli, okay, remember they have those membranous sacs. Under there, they have a cellulose-like material that makes these armored. So their alveoli have almost like cell wall material in it. But it's not a cell wall. Cell walls are outside of the plasma membrane. This is inside the plasma membrane, okay? So it's not a cell wall. All right, of course, being chrome alveolates, they're photosynthetic. They have chlorophyll A and B, A and C, and various types of reddish pigments that are called xanthophylls, right? And they store the food, excess food, starch. Some of these are symbiotic in corals. Corals are animals, okay? Corals are animals, and these are the endosymbionts that I was talking about, sometimes live inside of them. Certain species. However, these are not so attached to the corals that they can't live independently, okay? Because they do leave and you can find them both independent and in the coral. Interestingly, the shape of the cells are totally different when they're an endosymbiont versus free living. They completely change their gene expression, but we're still talking about the same species. All right, some of these produce red tides. In other words, if there's a lot of runoff into the ocean and there's a lot of nutrients there, these things, certain species will multiply very rapidly, and water turns sort of reddish color because of their pigments. Shellfish eat these things, and fish eat them, and soon what happens, if you've ever heard that they have a moratorium on eating shellfish off the coast here, it's because of this, okay? Because the toxins that these things produce accumulate in shellfish, and therefore they're toxic when humans eat them. And this red tides are starting to happen more and more often, probably because of human-made pollution. Okay? And Apicomplexa, that's also up here. Malaria parasite, okay, primarily, and these are all parasites, and they have a special structure on the cell that allows them to bore into the red blood cells, right? So this is for penetrating, and they're very small, very complicated life cycle. They are not photosynthetic, they're heterotrophic, but they do have plastids, indicating that they came from a photosynthetic ancestry. And their plastids are involved in fatty acid metabolism, something that's unique to them. So infectious stage in malaria, they can have many different life forms. Same species, different life forms. When it affects a human, the infectious stage is called a sporozoite. And their life cycle involves both sexual, asexual reproduction, both in their host and in an alternate host. They have usually two or three different hosts. For the malaria parasite, which belongs to the genus Plasmodium, it is transferred from humans to mosquitoes, but not all mosquitoes, only those mosquitoes of the genus Anopheles, okay? So other genera of mosquitoes will not carry the parasite. So around here we have a mosquito called Culex, widespread. We also have Aedes aegypti, which is widespread, and these do not carry they can't carry the parasite, only certain Anopheles mosquitoes. And it's only female mosquitoes that suck blood. Males do not. It's only the female that is sucking blood for her eggs, right? And so this is the parasite gets in the mosquito's salivary glands, and when the mosquito bites, it injects saliva into the wound because there's a painkiller in it and also an anticoagulant but also these malaria parasites. So you saw these slides. They reproduce asexually inside of red blood cells, and they're eating up the red blood cells. Once you have one infection, then all of these, their life cycles are synchronized in the same individual. And depending on the species of Plasmodium, they will all break out of red blood cells simultaneously, either on a 24, 36, or 48-hour cycle. And when that happens, the individual all of a sudden gets real sick, okay? Because they have all this broken blood cell debris in their body, and they suffer high fever, chills at the time. And then this is happening to them every, depending on the cycle, 24, 36, or 48 hours, depending on the species of parasite that they have. Okay. All right, the last group up here is Ciliophora. You just need to know them as ciliates. So paramecium that you looked at as ciliate, there's many of these. Their bodies are covered with cilia, okay? And you just need to know them as ciliates, but they have an interesting type of life cycle. They actually have two different forms of the two different nuclei, major nucleus and a minor nucleus. One of them, what's called macronucleus, controls the metabolism in the cell, and the micronucleus is only used for sexual reproduction. And they have a real different type of sexual reproductive cycle. Okay, so that covers most of them. What do we have here left? Okay, so that is all of the chrome alveolata, but in SAR, we have this one branch called rhizaria. Now, there are several things. The branch is called rhizaria, but within rhizaria, there is something called foraminifera. Okay, so there are other rhizarians that I'm not going to talk about. Okay, so I'm going to have two levels of synapomorphies here. One for rhizaria, and one for a subclade of rhizaria called foraminifera. Okay, so for rhizaria, these, in fact, all of these are primitively heterotrophic. All of them are primitively heterotrophic. That means they never have any evidence of having photosynthetic ancestors. Okay, just like excavata. Most of excavata is also primitively heterotrophic, with the exception of the euglenoids. Okay, that's the exception. All right, so so needle-like pseudopodia. They're amoeboid. However, they don't have lobe-like pseudopodia. Last semester you looked at an amoeba. They're in a totally different place. Okay, here in biology. These have needle-like projections. It's still called pseudopodia, and that's where they take in nutrients by endocytosis. Okay, one clade within rhizaria is foraminifera. You just need to know them as forams. Okay, forams. That works just fine. Okay, and they were the ones that you looked at on a slide yesterday. They secrete a calcium carbonate shell. It has many chambers. It looks like a miniature snail shell. However, these things are unicellular. They're real, just like one thing. So if you have a shell like that on the outside, how do you feed if you're a heterotroph? Well, that shell has holes in it, and the pseudopodia are sticking out through the holes. Okay, so they're still taking in nutrients that way. All right, now most of these are, well, they're all marine, but the vast majority of them that we know are extinct. Many of them went extinct at the end of the Mesozoic, along with most of a lot of dinosaurs. So they died in that particular episode. We still have a few of them today. They're all marine. So these cytoplasmic pseudopodia sticking through pores in the calcium carbon shell. Right? That's how they feed. They're all heterotrophic. 90% of what we know are extinct. All right. So that takes us over to our two last clades, supergroup clades, which we will expand on. So I'll just introduce them here. Oh, incidentally, yeah. Chalk. How many of you know the White Cliffs of Dover, England. You guys don't go. One person, good, okay. He gets out. The rest of you don't go anywhere. Have you ever been outside of Fremont? Have you been to Berkeley? All right, but anyhow, oceans, seas that had a lot of these in the past, when they die, this calcium carbonate accumulates in the sediment, and that's exactly where we get chalk from, okay? From these. Okay, supergroup Archaeplastida. For what we're covering in here, red algae, green algae, and plants. So they're the original primary endosymbiotic origin of chloroplasts. It happened once, okay? Happened once in there, after which, in the early days of their history, they evolved two different lineages, red lineage and a green lineage in their chloroplasts. Green lineage with chlorophyll A and B, red lineage with chlorophyll A and C. Okay? And the major clades that we will look at is Rhodophyta, red algae, Chlorophyta, green algae, and land plants. Those are going to be the major ones. They're going to be the subject of the next two lectures, right? Then our last one, Amorphia. Over here, separate supergroup. This here is diagnosed mostly, now I said defined. Change that to diagnosed, okay? Yours says defined, because really the phylogeny defines the group. But the synapomorphies diagnose the group. That's our evidence of their monophyletic relationship. Now this here has something unique, big word here, triple pyrimidine biosynthesis fusion gene. Okay, and that's the enzyme codes for. Let me just explain it. You just need to say a pyrimidine fusion gene, that's all. Okay, so what are pyrimidines? Okay, so it's a nitrogen-based single ring, okay, like cytosine and uracil, etc. But in the pathway to form those, there are individual enzymes. Okay, it's not just one step. Okay, now this here includes a structure, an enzyme that is the fusion product of three different genes. In other organisms, all other branches of eukaryotes and prokaryotes, these three genes are separate. They have their own promoters, their own enhancers. They code for monocystronic messenger RNA. Here in this group, they're fused together as one large supergene, okay? And it's pretty unique. They have one flagellum. Most of these have two or more. And in fact, two flagella in eukaryotes was probably ancestral because it is widespread, okay, having two. But here, they've lost one. So they only have one flagellum. All these are primitively heterotrophic. In other words, there is no evidence of any ancestry of photosynthetic organisms in this group. It is heterotrophic, completely. And the three major clades that we'll be looking at, something called amoebozoans. So this contains slime molds and lobate amoebae. The amoebae that you looked at in the lab last semester belong here, okay? They're not rhizarians, okay? And fungi is going to be another major group, and animals. These are the major groups that belong to Amorphia. And this will be the subject of future lectures, okay? All right, so these eukaryotic life cycles I'm going to not cover, okay, except for one. So you're not responsible for these, because we got to move on. I just want to cover just malaria, okay, just malaria. All right. So these are parasites. This is how I'm going to be writing the classification as a lineage. So this is not full. You'd have eukarya, SAR, chrome alveolata, AB complexa. Okay? And so you don't have to worry about phylum, kingdom, etc., just the lineage, okay, within each. So these parasites, get through this real quickly, and then I'll go over this again. The infectious cell is a sporozoite, and they have very complex life cycles. I'm only going to talk about Plasmodium. That's the genus that causes malaria, and there's 4 different species that cause it in humans. Spectre is Anopheles mosquitoes, female Anopheles mosquitoes, and nothing else. Okay, so this is how it works. First of all, a mosquito has to drink blood from an infected person. What the infected person has in their bloodstream are a form of the parasite called a gametocyte. These are haploid. They're the gametes. They're the gametes. And once it gets eaten, and the gametocytes fuse together to form a zygote, sexual reproduction. That's diploid. And that zygote is in the mosquito's gut, and it'll undergo meiosis. This is the membrane of the gut. They'll crawl out of the lumen and form what looks like a big blister, and they become sporozoites. They look completely different, okay? These sporozoites will go to all parts of the body. A lot of them will end up in the salivary glands. Then when the mosquito feeds on a human, they will be injecting the sporozoites into the wound. This gets into the bloodstream, and these will swim to the liver, where they infect liver cells and become a different form called merozoites. They look differently. The merozoites will reproduce asexually, eventually leave the liver, and then invade red blood cells, where they're feeding on hemoglobin, and they're dividing asexually. Asexually, then they'll burst out of the red blood cells and infect more red blood cells. Again, again, again. And as I said, they're all synchronized. They're all synchronized doing this at the same time. Some of these, usually in the evening, will develop into gametocytes. Why in the evening? Because that's when the mosquitoes bite. And otherwise, these are open to the immune system. The merozoites in the red blood cells, the immune system can't see them. They're protected, okay? But the gametocytes can be seen by the immune system, and so they're there just briefly at the same time that the mosquito's going to be feeding. And they're taken up then into the mosquito, and that completes the cycle. Now, actually, there's more forms than just merozoites, gametocytes, and sporozoites, but that's all I'm going to say. Okay? And that's it. We're done. There's a person swimming, and guess what's causing all that bioluminescence, blue? Dinoflagellates. Many of them are bioluminescent. And when you disturb ocean waters, you'll see this blue. I went swimming once in Jamaica, and it looked like that. Have any of you been to Jamaica? I shouldn't even ask that. Okay, so, okay, that's it.